there's not much need for statistical manipulation if you're not working with samples from the field, of course. For instance, if you're just interested in some kind of model, or if the data you have is not, if you're not interested in the biological variability, actually, so you're interested in just a model of something that you extracted from one lineage of cells that doesn't vary. So when you do that, theoretically, all the variation you're going to have is just technical. It's not biological. Let's say you could really do that. Okay, I can have a invariable cell, lineage of cells here, that they're all the same. I'm sure there's no variation. If I repeat the experiment 10 times, I'm going to have exactly the same result. I'm technically perfect, everything. What's the problem with that? I cannot infer that result to any other population of cells but the one you used. Because there is no biological variation accounted for. And biology varies. So if you want to make inferences about a group of people, not people, but any biological specimen, whatever you want to, cells or whatever, you need to take the variability into account. Otherwise, there's no way to infer that to that whole population where even that cell, those cells came from. But you're only talking about those set of cells, not about the set of cells that came from a different mouse or something like that, or whatever. You know. So, and this is one thing that I like to stress, that there are these two kinds of variability that we have to deal with. One of them I want to avoid is the technical variability. I don't want to perform an experiment exactly the same as the other one and find my measurement 10 times above the one from the other. Of course not. I need to replicate that very well. I don't want technical variability. And that I want to avoid. And in many instances, not only I try to avoid, but I tend to disconsider that error. Because again, it's something that we don't think much. But let's say I will, instead of the age of this group and this group, I'm going to have your weight. Okay? So I'm going to get a scale, right? And I'll ask everybody to step on the scale and get your weight. What's going to happen? I guarantee you that if the same person steps on that scale 10 times, I'm going to have at least 5 different measurements. And why is that? The scale also has a variation. Just test it in your home. Step on the scale 10 times and see if it's going to be exactly the same number every time you step on. No, it won't be. Which means that if I'm using weight as a variable and I'm using just one measurement, there is an error there, a technical error, but that I'm not considering, because nobody's going to weigh the patient 10 times and then get an average, right? I'm going to say, no, that's, I'm going to neglect it because it's a very small error. It's like, let's say, it's 10 grams in a 60 kilos, 60 kilograms. So I'm not going to worry about it. But even for a very simple variable like that one, weight, right? Very simple. But there could be a measurement mistake there, measurement error. So everything we measure, there is an error. And that's the error I want to avoid or control or get rid of it. The other error that biostatisticians, statisticians, and so forth call error, that's not really an error. That's an error for the model they're using to compare, for instance, the averages. But that's the normal distribution of that variable. So that's not something I want to get rid of. That's something I want to study. I need the variability. I need that kind of variability. Because if I'm going to make an inference about that population, and I know the population varies, my sample has to vary as well. If my sample is static, it doesn't vary, it's invariant. There is no way I'm going to make an inference about the population where that sample came from. Because I know it's not true. Right?